Hi, everyone. This will be a presentation on the 2013 research paper, Premature Aging in Telomerase-Deficient Zebrafish. So rather than beginning this presentation with a list of facts, I figured we would start with some questions, because questions are what progresses science, right? I know all of you watching will have questions of your own, but I figured I'd throw in some of mine, which encouraged me to write a paper about this topic, just kind of things I was thinking about. So we have, why do organisms age? Wouldn't it be more efficient for an organism to live forever? And can the phenomenon of aging be sped up or slowed down? So these are all good questions. The first two are definitely way too ambitious for the scope of either this presentation or my full review. Aging is a complicated process, but some factors that control it are actually pretty easy to manipulate and observe. In humans, there are a couple rare genetic diseases that are characterized by premature aging, like progeria and dyskeratosis congenita, for example. So, what's this paper all about? So, as it turns out, the current model, which is used for a lot of human diseases, mice, are really very imperfect and unsuitable for telomerase-centric treatments and studies. Um, Their telomeres are significantly longer than human telomeres. And in case anyone needs a refresher for what a telomere is, it's a supplemental region of DNA added on by a telomerase during cell replication. Telomeres get cut down in length a bit with each replication cycle, and when they run out, the cell dies through apoptosis. And in some species, physiological aging occurs. So what else do we have going on in this article? Telomerase deficient zebrafish have been shown, P53 dependent premature aging and reduced lifespan in the first generation, which reflects the similar telomere length in fish and humans. And inhibition of P53 rescued the adverse effects of telomere loss, indicating that the molecular mechanisms induced by telomere shortening are conserved from fish to mammals. So the main idea here again is that zebrafish are an ideal model for research into telomerase biology and telomerase specific disease as they recapitulate human uh, mechanisms. They're useful for the discovery of new treatments and able to temporarily reactivate telomerase expression in individuals with dyskeratosis congenita. So for the purposes of this paper, um, the researchers, the most important thing they did was that they created a variant line of zebrafish which had telomerase reverse transcriptase knockout, and they observed the effects of that. So let's go into the results. So part A here is a representation of the survival of three genetic backgrounds. The time post-hatching is shown in weeks on the x-axis, and the survival rate is on the y-axis. The colored lines represent different genotypes. Blue is TERT wild type, red is TERT knockout, and green is heterogeneous. TERT knockout zebrafish showed a reduced median lifespan, around 67 weeks, compared with TERT heterogeneous at 105 weeks, and TERT wild type at upwards of 110 weeks. So TERT knockout had a greatly reduced lifespan as compared to the wild type or the heterogeneous versions. Part B here on the left is an image of a TERT wild type on the top and a TERT knockout zebrafish on the bottom, both at 11 months old. Researchers made note of spinal curvature, liver and retina degradation, and infertility. These are all hallmarks of advanced age, even though both of these zebrafish are only 11 months old. So it just goes to show the impact that telomerase reverse transcriptase knockout can have on the physiological level. Let's move on. Part C here shows stained liver sections showing cytoplasmic vacuolization of hepatocytes and were pass positive. That's marked with white asterisks. I'm not sure if you are able to see it in the resolution of this image, but that would be observable in the TERT knockout, those top two columns on the right. Right column, top two boxes, top and middle box. H and E stained eye sections on this bottom row revealed a decreased thickness of both retinal pigmented epithelium and the photoreceptor layer, which had a greatly atrophied appearance. So in this bottom row here, on the left bottom picture, we have the wild type with those two retina layers marked with that white bar. And in the heterogeneous uh, genotype, you can see it looks pretty similar, but in the knockout wild type on the right on the bottom, that retina is just so much more degraded. It's so much thinner. So this is that retina degradation that they observed in the TERT knockout. Moving on, part A is a representation of testes cell distribution from the wild type on the left side and the TERT mutant genotype on the right, according to their telomere length. Medium fluorescence intensities indicated for each genetic background. The telomere length testes cells from knockout zebrafish suffered a decline at 8 months old. So if you just follow my mouse here in the wild type, you can see that the telomere length is maintained. It's increased at 4 months, 8 months, 18 months. Whereas in the TERT knockout here, there's a notable decrease from 4 months to 8 months and still within that range at 18 months. Part B here on the right is an image of zebrafish testes samples from TERT wild type and TERT knockout backgrounds at 4, 8, and 18 months old. Asterisks in panel 6, if you can see them here, indicate atrophic tubules, and arrowheads indicate atrophic stroma. Again, this is another hallmark of aging. Both 4-month-old specimens and testes tissue samples from TERT wild type and TERT knockout zebrafish showed similar morphology, as we can see, in terms of the structure of seminiferous tubules and normal connective stroma. At 8 months, TERT wild type presented the same morphology. Sorry, I'm having trouble tracking my mouse here. Okay, I see it. Here at 8 months, TERT wild type presented the same morphology at 4 months of age, and this continues to 18 months of age. Sorry, where'd my mouse go? Okay. It's very comparable as this organism ages, whereas in the TERT knockout version, you just see this this atrophication of the area. So in telomerase deficient testes, you're seeing the development of a lot of apoptotic cells. These results correlate well with the decrease in telomere length, and they suggest a direct link between telomerase function, fertility, and aging. So let's recap the findings in this paper. Symptoms, physiological symptoms of spinal curvature, liver and retina degradation, and infertility were observed. And note, this is something that you could consider to be a hallmark of aging in both zebrafish and humans. So this just, again, connects the model, the usefulness in zebrafish as a model for telomerase-related disease research. They found that second-generation embryos died in early developmental stages. Restoration of telomerase activity rescued telomere length and survival, which indicates that telomerase dosage is crucial. And these results point to the usefulness of the zebrafish model for the discovery of new drugs able to reactivate telomerase in individuals with dyskeratosis congenita. So some future applications. The TERT-deficient zebrafish model described in the study could be an excellent model for investigating the role of alternative lengthening of telomeres in telomere maintenance in the absence of telomerase activity. And overall, there are therapeutic benefits of new drugs that can induce the expression of the wild-type TERT or TR allele in heterogeneous DC patients. And this line recapitulates the mechanisms of age in telomerase-deficient mice and individuals with DC. Again, zebrafish are an excellent model for this particular genetic disease. So here is the paper itself cited, and I look forward to reading and replying to everyone's questions. And thank you very much for watching.